Hello, welcome everyone to this interview. Um, First of all, I want to thank Professor Greg Dussor and Dr. Philip Holland for joining this interview. Professor Greg Dussor works in the University of Texas in Dallas, and Dr. Philip Holland works in King's College London. I want to welcome you to this interview where I would like to talk about the translation and impact of current animal models.
So to begin with, as a researchers, I think it's common to hear people approaching you during congresses or stuff like that, asking, but do mice have headache? Do rats have a headache? So to put sort of everyone in the same page, I wanted to ask you, what do you model as leading researchers in the field when it comes to studying migraine in animals? So what are the questions you are trying to answer when you are studying the different or using the different models of migraine?
Yeah. So I guess maybe before the answer to that question, can they have a headache? Is it possible for them to have a headache? And if we agree that maybe the relevant anatomy that would be necessary to have a headache is nociceptors that innervate the meninges covering the brain, they certainly have those. They have meninges and they have nociceptors that innervate the meninges. So conceptually, they could have a headache, but do they? It's almost impossible to know that in an animal. So we try to be careful not to say that they're having headaches when we give presentations or we write publications. What exactly is it that we are modeling? We say hypersensitivity. Periorbital hypersensitivity is often what we're testing. Bonfry responses to tactile stimulation of the forehead of the animal or maybe other parts of the body. Is that actually headache? I think it's a good question. And because it's difficult to know whether they are having the sort of unilateral throbbing pain that would be characteristic of something like a migraine, we try not to use that word because it's just, we don't think it's appropriate to use that word. So, Phil.
Yeah, no, I fully agree. A great answer from Professor Dussor there. And I think one things we do is we use a lot more electrophysiology in our lab. So we're actually recording from these nociceptors directly and looking at their activation. But again, there's this complexity between what is nociception and what is pain. And pain is such a cognitive process. And again, we'll never be able to know whether, for example, a mouse or a rat is having that, complex features of what pain is with the emotional and the fear components and the different aspects related to that. But we can tell that those nociceptive neurons that are processing this sensory information from the meninges, whether they're activated or not, and importantly, whether, for example, new therapies will be able to block that response or not. And I think that really gives us a really important clue into whether they're very likely to block those responses in humans as well. Beautiful study recently by Professor Will Rental, for example, showing that similarity in transcriptomic profile between human and mouse neurons, for example. So it really gives us a really strong platform, I think, to build from.
Thank you. So, for example, when you're using an animal model, which readouts are you looking for? So you just mentioned, you talk about hypersensitivity, you talk about activation of the vascular system. The question, I guess it will be like, why? Like, do you want to model a specific aspects in patients such as allodynia, or are you using that as a readout of having a headache?
Yeah, I mean, because of the difficulty in determining whether or not the animal is having a spontaneous headache, we have to use these other sort of associated features. I think we and others have tried to do a good job in the field of translating back from humans into the animals endpoints that we at least know are present in the human. And so if we're going to test periorbital allodynia as an endpoint, does that actually happen in people, in humans that are having migraines? And yes, it does. If we're going to try to test something like a change in locomotor activity or behavior, we know that headaches in humans are aggravated by physical activity. And so if you see a change in physical activity in the rodent, you're at least using an endpoint that we know has some relevance, some translational relevance to humans. Some of the other endpoints that we've tried to use, grimace, looking at facial expressions of the animal, trying to sort of read into what the animal might be feeling by what the expression is on its face. Do we use the grimace? Do we use grimacing in humans? We don't need to use grimacing in humans. You can just ask the human whether it's in pain, but humans grimace when they're in pain. And so I think the key thing that we always try to do in the animals is make sure there is some human relevance and human connection to the endpoint that we're trying to use. Otherwise, yeah, we may be studying something that is completely irrelevant for the human disease.
Yeah, no, definitely. I think we're looking for something that's physiologically and functionally related to the human condition, and not something that everyone does, but a very obvious example of this is, for example, the recording of cortical spreading depression as the correlate of migraine aura. I mean, it's extremely difficult to properly measure cortical spreading depression in the human brain because it's an invasive procedure. And we know that these spreading depolarizations happen, for example, in brain injury and trauma. So we've been able to do that, but we've never recorded them truly in the sort of what we're looking for, the uninjured human brain. We have some imaging surrogates that they do happen, of course. Now, in the animal, of course, we can really record that feature and see that spread across the cortex. The question is, would an animal then have the same physiological response to that aura that you would see in a human? Because, for example, the rodent cortex is, mesencephalic. It's smooth compared to the highly convoluted and folded nature of the human cortex, with much more sensory kind of processing going on there.
And I would say, as many nights as I've laid in bed trying to figure out how to measure an aura in a rodent, I have not come up with a way to do this yet. So if anyone has any good ideas of how to tell whether a rodent is having an aura, please let me know.
But do you think then for aura it would be wise to use animal models that are not lysencephalic? So should we go back to cats, or should we try? I've seen more in stroke research. I've seen that they use goats, for example, or sheep. So do you think we should move to eurencephalic brains for that?
Yeah, I think we have to accept that there are limitations in what we do. And of course, with all animal research, what we're looking to do, our preclinical research is, does this drive towards the least sentient species where, to do as little harm as possible and make sure that we're refining and making our processes as, ethically sound as possible. We could turn that round and say, for example, well, rodents are nocturnal. And the humans are diurnal. So, there's a specific hamster, for example, I think it's the Syrian hamster, that is diurnal like humans and not nocturnal. So there are a number of things there that we really could be looking at. And I guess it depends on the question you're wanting to ask, and the detailed nature of that question. But I will say that actually, rodent research really has, really stood up in terms of migraine research and other headache research, and allowed us to make some real fundamental breakthroughs that have made a difference translationally. So I think we're on the right path, but yes, these models can always be refined.
Continuing with the refining, actually, more recently you hear people saying that we should get rid of animal models and have more human-based approaches like organ on a chip and so on. What are your thoughts on that? Do you consider it is possible actually to get rid of these animal models, or should we always use animal models for research?
Yeah, I'll start, I guess. But I think it's always important to use human whenever possible. So one of the examples that we have been trying to incorporate over the last few years is to incorporate human tissue from organ donors. So to the extent that you can learn things about neurons, that trigeminal neurons, for example, ex vivo or in vitro, by taking human tissue and studying it in a dish or outside of the body, where you can't do these experiments in the body in a human, those kinds of studies are valuable. And so to the extent that we can be using human tissue in experiments, I think we should. But the limitation of that is even if you got human trigeminal ganglia, which we are doing now, and you put those in a dish, and you can literally study human neurons in ways that you can't do in the intact human. In the dish, you don't have a circuit. So those neurons are very valuable in certain kinds of things that they can tell you, but they're not connected to anything anymore. So we don't know how they integrate themselves into the rest of the nervous system and how they might respond to the surrounding environment in the meninges, for example, because they're no longer in the meninges. So I think that there's always going to be things that animal models are going to be capable of letting us know and ask that are just never going to really be possible to do in humans.
And I think building from that, it's really a great answer actually. But there are a number of things that, and you made a great point earlier on, that a lot of what we do in the animal is reverse translated from the human, and I think that's the starting point. If we're going to use an animal model, it has to be anchored in a relevance of the human condition that we're using. And that's a key point. And there's a scope there for fundamental research as well, that really, really early level science that allows us to drive discovery before we get to humans. in terms of, screening molecules, safety, outcomes, target validation that's really important here. And I think, we mentioned this comment about, well, can you model, these things in a dish, or could you use organoids? And we've made huge strides in that area, actually, and Professor Dussor's doing some wonderful work there. But what I think we have to remember is that migraine and many of these other conditions are a whole brain condition, the peripheral, the central nervous system, those connectivities. And, it'd be wrong of us to say that we fully understand that connection, but actually we have to study that in the connected system. And at the moment, that connected system is only available in a living organism.
Yeah.
And therefore, that takes us back to where needed an animal model of, say, migraine, cluster headache, or systemic conditions.
Now that you mentioned cluster headache, what do you think we are missing in the headache field when it comes to animal models? Because usually when we talk about animal models of headache, we think about migraine. But what do you think we are missing in the field, and what do you think we should have in the future? So what should we develop?
Yeah, I think we sort of gave away the punchline there. We need an animal model of cluster headache.
Yeah.
There's plenty of other headache subtypes that it would be nice to have animal models of as well. The caveat to that is that coming back to earlier, the difficulty in knowing whether or not an animal is actually having a headache, even if you were confident that the animal was having a headache, what kind of headache is it actually having? What does it feel like to the animal? Is it unilateral, which can be difficult to tell depending on how you might be measuring that endpoint? Is it a cluster headache-like sensation that the animal is experiencing? So yeah, we absolutely need models of more subtypes of headaches, but the challenge is still going to be determining what exactly it is that animal is experiencing. Now, if there were specific features of cluster headache that were unique to cluster headache that you could backward translate into the animal for a behavioral model, for example, that might be valuable, because then it would sort of tie the model specifically to something that is a feature of cluster headache, like we've tried to do with migraine. There are many subtypes of headache that don't really have cutaneous allodynia with them. Migraine does, and so that endpoint is a valuable translational feature. If there were things like that about cluster headache that we could bring into an animal model and give it some specificity to that particular condition, that would be super valuable.
Yeah. And I think, to be open and honest and say that's actually something we're working on actively, certainly within our laboratory. If we think about migraine, we spoke about allodynia, we spoke about these other features, but for example, Professor Andy Russo pioneering photophobia and different sensory sensitivities. So taking associated phenotypes with that disease and moving them into a valid model. So the existing model for cluster headache at the moment, for example, relies on triggering this parasympathetic trigeminal autonomic reflex. And in the rodents, we can see lac formation and we can see features that happen in the humans, and that's great. But where these models are lacking, for example, is that they're not behaving animals. So we don't see these associated features of agitation and restlessness that you might see in your patients if you're seeing this condition. And I think over time, these models will be refined and developed. And we've got new techniques now. For example, Professor Dussor gave a lovely teaching course here on chemogenetics and the ability to now to be able to manipulate these networks in rodents. And I think that's where preclinical research will really be taking us forward now. We can start to interrogate specific circuits and look for phenotypes that are similar to the clinical condition that might then complement human imaging that has given us a clue, but again, at a much larger scale where we can't see the specifics of those specific nuclei or what they're actually doing.
Is there anything that you would think, besides something that we're missing when it comes to modeling different headache types or modeling different phases of a migraine, is there anything else you think we should be thinking about in the future when it comes to animal models? Like any considerations we should have, for example, when it comes to studying the different aspects of primary headache disorders or secondary headache disorders, for example?
Yeah, that's a great question. I mean, I think there's a lot of things that you could say here. One of the limitations of mice and rats is they don't vomit. So they don't have the relevant anatomy to be able to vomit. And given that that's a prominent feature of many headache subtypes like migraine, would we know that they are having nausea-like symptoms? That'd be difficult to know. Again, the aura is difficult to tell whether that's actually happening in the animal. The other thing that I don't think that we as a field have been really good about in working with animal models, and certainly with novel compounds and novel sort of approaches that might be therapeutics in the future is really assessing what the side effects of those treatments might actually be. We always focus on the main therapeutic effect, on the pain-like response or the photophobia-like response, but we don't necessarily do a good job of asking, well, is this intervention that we're doing that might work for photophobia or the allodynia, is it actually elevating the blood pressure too much? And so that might end up being a problem that would never move forward in humans. And we don't ask those questions very often. And I think we should do a better job of that kind of thing.
I certainly think that's one of the issues with behavioral models. One of the things with the detailed electrophysiology we do is, again, we do model often blood pressure, but again, it's at a very acute setting, not often in that chronic setting that many of these treatments are being given. And I think for me, if I could probably say one thing that we could do, and I mean like more immediately that we could really make a change, I think it's a bit of a standardization across the field. We have now a number of different models of headache. So if we could actually think about whether it's for drug development or drug testing, but actually really having a sort of a validation pipeline of what we feel as an expert consensus panel across not just ourselves, but throughout the scientific community, what are the correct models that we really should be using to validate a new compound? And therefore, compounds coming along in the future, for example, can then be validated in that same battery of tests, and we get that relative response. At the moment, it can be a little bit varied in terms of which tests we're using.
Or even within the same test, I don't feel like there's a proper consensus when it comes to some people use some people use, I feel like every group has their own recipe of what it's added, and also every group has their own recipe of how to do photophobia, how to do it. I feel like there is not a single specific way of running one particular experiment.
Yeah, and I think that speaks to, this is not a headache problem. This is a science problem across the board, and it speaks to reproducibility. And I think ultimately, if we're going to be validating these works and taking compounds forward to human trials, whether it's validating side effect profiles or validating therapeutic efficacy and response, we really want to think about that validation of those models and being able to reproduce that. It's simply not enough that one lab in one place shows a response, but actually we saw in this meeting with some of the stress priming models that actually Professor Dussor spoke about, that they've actually now been replicated, these sex differences, for example, in response to CGRP, they've been validated in 2 or 3 different laboratories now, and I think that's really important that we need to get there.
Yeah, but what would be really nice to have is a headache research academy where, we have everybody who's going to be doing this certain kind of behavioral test. They get together and they agree on some fundamental features of how these things are done, and maybe trainees are part of this opportunity and they learn as a group how to do this, so it's at least standardized across the field. But part of the problem with this is it's hard to get funding. It's hard enough to get funding as it is. It's somewhat boring but important to propose that I want to try to replicate what Dr. Holland showed in his lab. It's difficult for me to get funding for that kind of thing because it's not exciting enough, and that's an unfortunate situation.
Thank you so much for being part of this interview. I really hope that the IHS takes into account the Headache Research Academy. I think it will be an excellent idea. And without further ado, I again want to thank you for this interview.
Thank you. Thanks for the great questions.
Thank you very much. Great to be talking.
Thank you.